Welcome to Plunge into Science. Today, we are not just dipping our toes into the water. We are embarking on a journey to the most extreme, most alien, and most inaccessible location on our own planet. We are descending into a world of absolute darkness, crushing pressure, and life forms that defy our very understanding of biology. We are going to the Mariana Trench. This isn't just a trip to the bottom of the sea. It's a journey to the bottom of what's possible. So take a deep breath of air while you still can. Because where we're going, there is no surface, there is no sun, and the weight of the world will be, quite literally, upon us. Section 1. The invitation to the abyss. Before we begin our descent, I want you to do something for me. Close your eyes, I mean it. Just for a moment. Picture the world's most formidable mountain, Mount Everest. Imagine its snow-capped peak, the thinness of the air, the sheer staggering scale of it. It's a symbol of ultimate terrestrial achievement. Now, in your mind, take that entire mountain, all 8,848 meters of it, and flip it upside down. Plunge it into the ocean. Imagine its peak pointing down, down, down into the blue darkness. Even after all that, you would still have over 2 kilometers of water, over a mile of nothing but deep, dark ocean above its submerged summit before you reach the surface. That is the scale we are dealing with. That is the Mariana Trench. It's not a pit, not a hole, but a colossal scar on the Earth's crust. A crescent-shaped canyon over 2,500 kilometers long. This is a place so remote that more people have stood on the surface of the moon than have visited its deepest point. So, why do we feel this pull? What is it about these absolute extremes that captivates the human imagination? Is it the same drive that pushes us to summit mountains or explore outer space? Perhaps it's a more fundamental curiosity. We stand on the shores of the ocean and see a vast, shimmering surface, a mirror to the sky. But we know intuitively that this surface is a veil. It conceals the largest living space on our planet, a three-dimensional world that makes up over 95% of the biosphere. And the Mariana Trench is its ultimate final mystery. It's the basement of the world. What secrets are locked away in a place defined by the complete absence of everything we consider normal? No light, negligible warmth, and a pressure so intense it can warp steel. The questions are intoxicating. What kind of life could possibly exist there? And more profoundly, how? How does biology, in its infinite creativity, solve the problem of existence at maximum pressure? This journey is our attempt to find the answers. Are you ready to leave the sun behind? Section 2. The geology of a plunging world. To understand the life in the trench, we must first understand the trench itself. It is not a passive feature. It is an active, violent, and incredibly slow geological event that has been unfolding for millions of years. The Mariana Trench exists because of a process called subduction. Imagine 2 colossal conveyor belts on the Earth's surface. These are the tectonic plates. The Pacific Plate, a dense, ancient slab of oceanic crust, is moving westward. It collides with the much smaller, younger Mariana Plate. But this isn't a head-on car crash. Because the Pacific Plate is older and colder, it's denser. So, when they meet, it doesn't crumple upwards to form mountains. Instead, it is forced to bend and plunge downwards, diving deep into the Earth's mantle. This downward plunge, this slow-motion geological bow, is what creates the trench. It is a wound where the skin of our planet is folding in on itself. Think about the forces involved. The friction, the heat, the sheer gravitational pull dragging an entire section of the ocean floor into the planet's molten interior. This process is not quiet. It is the source of the region's intense volcanic activity and earthquakes. The ring of fire, which encircles the Pacific, is a direct consequence of subduction zones like this one. But here the process is at its most extreme. The deepest point, the Challenger Deep, is the focal point of this immense geological strain. Now, consider this. What does that mean for the environment at the bottom? It means the trench isn't a sterile, static V-shape in the mud. It's a dynamic environment. There are seamounts, ridges, and valleys within the trench itself. More importantly, this geological activity creates chemical reactions. As the plunging plate grinds downwards, water is forced into the rock, reacting with minerals under intense heat and pressure. This process, called serpentinization, can release chemicals like hydrogen and methane. Why does that matter? Because those chemicals are a form of energy. In a world with no sunlight for photosynthesis, Could this geological energy be the spark that fuels a unique, hidden ecosystem? Could the very violence of the trench's creation be the reason life can exist there at all? Section 3. The tyranny of pressure. Let's talk about the single greatest defining feature of the Mariana Trench, the pressure. It's a concept we understand intellectually, but it's almost impossible to grasp viscerally. At the surface, we experience one atmosphere of pressure. It's the weight of the entire column of air above us, and we are perfectly adapted to it. Our bodies push out with the same force that the air pushes in. Now let's descend. For every 10 meters you go down in the ocean, the pressure increases by 1 full atmosphere. At the bottom of a typical swimming pool, it's barely noticeable. At the limits of recreational scuba diving, around 40 meters, it's significant. But we are going to the Challenger Deep, nearly 11,000 meters down. At that depth, the pressure is over 100 times greater than at the surface. It is more than 16,000 pounds per square inch. To put that in a mind-thinking way, imagine the entire surface of your body. Now imagine a large male elephant standing on every single square inch of it. No, that's not quite right. Imagine a stack of 3 large SUVs balanced perfectly on your thumbnail. That is the force we are talking about. It is a force that doesn't just crush, fundamentally alters the rules of chemistry and biology. So, what would happen to us or any surface creature at that pressure? First, any airspaces would be instantly and catastrophically eliminated. Our lungs would collapse into little more than a dense, fleshy residue. The gases in our blood would be compressed and forced into solution, leading to lethal nitrogen narcosis and oxygen toxicity. But it gets more fundamental. Pressure physically squeezes water molecules closer together, changing the way they interact with other molecules. The delicate three-dimensional folded shapes of our proteins, the very molecular machines that make our cells work, would be distorted, unraveled, and rendered useless. The fluid membranes that enclose our cells would be compressed until they become waxy and rigid, unable to transport nutrients in or waste out. Life as we know it would simply cease to function. So the question isn't just what lives down there. The question is, how is life even possible? How do you build a cell that can operate when the universe itself seems to be trying to squeeze it out of existence? Section 4. The descent through layers of darkness, our journey downwards is not instantaneous. It is a passage through distinct and increasingly alien worlds. We begin our plunge from the sunlit surface, the epipelagic zone. Here, in the top 200 meters, is where all the familiar marine life thrives. Photosynthesis reigns supreme. The water is warm. The colors are vibrant. But as we sink, the light begins to fail. We cross the 200-meter mark and enter the mesopelagic zone, the twilight zone. It lasts down to about 1,000 meters. The sun is now a faint, ghostly blue glow far, far above. There is not enough light for photosynthesis, but there is enough to see. And the creatures here are masters of that twilight. This is the realm of bioluminescence. Fish with massive, hypersensitive eyes hunt for prey that flashes with its own living light. Many creatures are dark red or black, colors that are effectively invisible in the dim blue light. Others are transparent, like ghosts in the water. We are sinking past lanternfish, vampire squid, and strange gelatinous siphonophores. The pressure is already immense, up to 100 atmospheres, enough to crush a military submarine. The temperature has plummeted. Then at 1,000 meters, we cross a profound threshold. We enter the bathypelagic zone, the midnight zone. The last photon of sunlight from the surface is gone. From here on down, there is only one source of light, the light that life itself creates. The darkness is absolute, eternal, and unchanging. We sink deeper, past 4,000 meters into the abyssopelagic zone, the abyss. The water temperature is now just a few degrees above freezing. The pressure is over 400 atmospheres. The creatures here are sparse, strange adaptations to a world of scarcity. We might see the eerie anglerfish, with its glowing lure, or the gulper eel, a creature that is mostly mouth, ready to swallow anything it encounters. Finally, below 6,000 meters, we enter our destination, the Hadal Zone, named after Hades, the underworld. This zone is comprised solely of the world's deepest trenches. We are now in the Mariana Trench proper. The pressure is astronomical. The cold is relentless. The darkness is primeval. We have left the world of the sun entirely behind. The rules have changed. What does it feel like to exist in a place that has never, in the 4.5 billion year history of the Earth, seen a single ray of sunlight? What does that do to the rhythm of life, to the very concept of day and night? We are now truly in another world. Section 5. The Vessels of Discovery. How do we even peer into this realm? We can't just send a person in a diving suit. We can't even send a standard submarine. Visiting the Hadal Zone requires technology at the absolute cutting edge of material science and engineering. It's a history of human ingenuity against unimaginable physics. The story begins in 1960 with the bathyscaphe Trieste. This was less of a submarine and more of an underwater balloon. A massive, thin-walled float full of gasoline, which is lighter than water and incompressible, was attached to a tiny, monstrously thick steel sphere for the two-person crew, Jacques Picard and Don Walsh. The sphere's walls were 5 inches thick. They descended for nearly 5 hours, hearing unnerving creaks and groans as the vessel compressed around them. When they landed on the bottom, in a cloud of fine diatomaceous silt, they became the first humans to reach the deepest point on Earth. What did they see? They reported seeing a flatfish, a soul-like creature, a claim that has been debated ever since due to the pressure effects on vertebrate bones. But their achievement proved it was possible. For over 50 years, no human returned. The challenge was too great. Instead, we sent robotic explorers. The Japanese probe Kaiko made several trips in the 90s, collecting the first samples of Hadal organisms. The American hybrid ROV Nereus explored the trench in 2009, but was tragically lost on a later mission. Then in 2012, filmmaker and explorer James Cameron made a solo descent in the Deep-Sea Challenger. This was a radically different design, a vertical torpedo built to sink and rise quickly. He piloted it from a cramped 43-inch sphere, surrounded by cameras and collection arms. He described the bottom as lunar, desolate, and isolated. He was the first person to truly spend time there, filming and observing. What do these missions tell us? They tell us that exploration at this level is a razor's edge between success and catastrophic failure. Every weld, every seal, every line of code has to be perfect. The pressure is an unforgiving adversary. A microscopic flaw in the steel of a cryosphere would cause an implosion so instantaneous and violent that the water rushing in would heat to the temperature of the sun's surface for a microsecond. Why do we take such risks? What is the value of sending a person versus a robot? Is there something irreplaceable about having a human consciousness bear witness to such an alien place? Section 6. The Ethereal Mariana Snailfish. Let's meet one of the undisputed kings of the abyss, a creature that embodies the triumph of biology over physics, the Mariana snailfish, Pseudoliparis swirei. Forget the image of a fish that you hold in your mind. This creature is not sleek, not scaled, not muscular. It is ethereal. It's a small, tadpole-shaped fish, about the length of your hand. Its skin is translucent, with no pigmentation, giving it a ghostly, pinkish-white appearance. You can see its internal organs through its skin. It has no scales, which would be useless and crushed under the pressure. Its skeleton is not made of hard, calcified bone, but is primarily cartilage, allowing it to be flexible and resilient rather than brittle and breakable. It appears fragile, almost unfinished, like a half-formed ghost, but this apparent fragility is its greatest strength. It is the deepest living fish ever discovered, thriving at depths of 8,000 meters. How does it do it? The secret lies in its chemistry. The crushing pressure of the deep sea actively works to deform the proteins essential for life. To counteract this, the snailfish floods its cells with a special molecule called trimethylamine N-oxide, or TMAO. You might know this molecule by its smell. It's what gives decaying fish their characteristic odor. But in the deep sea, it's the elixir of life. TMAO is a piezolite, meaning a pressure-protecting molecule. It works by supporting the structure of proteins, essentially creating a molecular scaffold around them that prevents them from being squeezed out of shape. The snailfish has the highest concentration of TMAO ever found in a fish. It doesn't just tolerate the pressure. Its very biochemistry is optimized for it. Here's the fascinating twist. This dependence on pressure is a two-way street. If you were to bring a Mariana snailfish to the surface, it would die. Without the extreme external pressure to counteract the high internal concentration of TMAO and other molecules, its cellular structures would essentially fall apart. It is a true piezophile, a pressure lover. It needs the crush of 800 atmospheres to live. Think about that for a second. We see pressure as a destructive, hostile force. But for this creature, it is the very thing that holds it together. It is a fundamental life-sustaining component of its environment. Doesn't that completely flip our perspective on what is normal or hospitable? What other fundamental forces of nature might life, somewhere in the universe, have co-opted for its own survival? Section 7. The Giants of the Hadal Zone. When we think of life in an environment of extreme scarcity, we often imagine things getting smaller, more efficient. But the deep sea has a surprising tendency: abyssal gigantism. And in the Mariana Trench, the star of this phenomenon is a crustacean, an amphipod. On the surface, you might know amphipods as scuds or sand fleas, tiny shrimp-like creatures, usually no bigger than your fingernail. But in the Hadal Zone, we find Alicella gigantea, and it lives up to its name. These are supergiant amphipods, pale, armor-plated creatures that can grow to be over 30 centimeters long, the size of a dinner plate. They look like something from a science fiction film, a prehistoric combination of a shrimp and a woodlouse. Why do they get so big? The science isn't settled, and that's what makes it so exciting. There are several leading theories. One is Bergman's rule, which suggests that in colder environments, a larger body size is advantageous because it means a lower surface area to volume ratio, making you more efficient at conserving heat. While the deep sea is cold, these are cold-blooded creatures, so that might not be the whole story. Another idea relates to the scarcity of food. In a feast or famine world, being big means you can eat bigger windfalls when they arrive, like the carcass of a whale or a large fish. A giant amphipod can tear off and consume huge chunks, and its larger body can then store those calories for a much longer time, allowing it to survive the long periods between meals. But there's a more intriguing theory related to the pressure itself. The immense pressure may suppress metabolic rates. A slower metabolism can lead to a longer lifespan and a delayed onset of sexual maturity. This extended growth period could simply allow the amphipods to grow to a much larger size before they stop. So the very environment that seems so hostile might be indirectly granting them this gigantic stature. They are the scavengers, the cleanup crew of the abyss. Swarms of them have been filmed descending upon bait, devouring a whole mackerel in a matter of hours, a frenzy of activity in the silent dark. What does it tell us? That one of the most successful strategies in this environment is to become a giant, armored scavenger. It tells us that the Hadal food web is built on the echoes of the world above, on the things that die and sink, a constant, gentle rain of sustenance we call marine snow. Section 8. Marine snow and the Hadal food. Web in a world without sunlight, there is no photosynthesis. So what forms the base of the food web? The answer comes from above. Imagine a continuous, gentle, and almost invisible snowfall. But this snow isn't made of frozen water, it's made of organic detritus. It's a mix of dead plankton, fecal pellets, shed exoskeletons, dust, and all the tiny fragments of life from the sunlit waters, thousands of meters above. This is marine snow, and it is the lifeblood of the deep ocean. As it sinks, this snow is a floating buffet for the creatures of the twilight and midnight zones. Much of it is consumed on its long journey down, but a small fraction, perhaps only 1 or 2%, completes the epic, weeks-long descent to the floor of the Mariana Trench. Once it arrives, this fine dusting of organic matter becomes the primary food source for a vast community of bacteria, foraminifera, and other deposit feeders. They are the grazers of the abyss, consuming this nutrient-rich mud. These smaller organisms are then eaten by slightly larger creatures, like the giant amphipods we just met, or the ghostly snailfish that drift just above the seafloor, hunting for them. But the food web isn't just this slow, steady drizzle. It is punctuated by massive, unpredictable windfalls. A whale fall. When a great whale dies in the open ocean, its colossal body, rich in oils and fats, sinks to the seafloor. This event is a jackpot for the deep-sea community. It is the equivalent of 1,000,000 years' worth of marine snow arriving all at once. First come the mobile scavengers, hagfish, sleeper sharks, and swarms of giant amphipods, who strip the flesh from the bones over months or years. Then come the enrichment opportunists, creatures like bone-eating ossidax worms, which colonize the skeleton, their strange root-like structures burrowing into the bone to consume the lipids within. Finally, years later, when only the barest minerals remain, chemosynthetic bacteria take over, metabolizing sulfides released by the decaying bones and forming the base of a new, localized food chain that can last for decades. Single whalefall can become an oasis, a multi-generational island of life in the abyssal desert. Doesn't this put a new perspective on death? In the deep sea, a death in the world above is not an end, but a profound and life-giving beginning for an entire ecosystem below. Section 9, the unseen kingdom of microbes. We've talked about the giants and the ghosts of the trench, but the true masters of this realm are invisible to the naked eye. The vast majority of the biomass and biodiversity in the Hadal zone is microbial. The mud of the seafloor is not just inert sediment. It is a living, breathing community of bacteria, archaea, and viruses, existing at densities of hundreds of millions of cells per cubic centimeter. These are the piezophiles, or barophiles, the true pressure lovers. Unlike the snailfish, whose proteins are merely protected from pressure, many of these microbes have enzymes that function optimally, or only under immense pressure. At surface pressure, their cellular machinery would fall apart. What are they doing down there? They are the ultimate recyclers. They consume the marine snow, breaking down complex organic compounds that are indigestible to larger animals. But more than that, they are the foundation of chemosynthesis. Remember the geological process of serpentinization we discussed? At vents and seeps on the seafloor, geologically produced chemicals like hydrogen sulfide and methane leak out. These microbes have evolved the incredible ability to use the chemical energy in these compounds to create organic matter, in much the same way plants use sunlight. They are the primary producers of the deep, a foundation of life built not on light, but on the Earth's internal chemistry. This discovery has profound implications that extend far beyond the Mariana Trench. It tells us that life can exist in the complete absence of a sun. This opens up the tantalizing possibility of life on other worlds. Think of Jupiter's moon Europa, or Saturn's moon Enceladus. Both are believed to have vast liquid water oceans hidden beneath a thick crust of ice. They have no sunlight, but they likely have hydrothermal vents on their seafloors, powered by the gravitational squeezing of their parent planets. Could there be chemosynthetic ecosystems thriving in those dark alien oceans right now? The study of the microbes in the Mariana Trench is not just deep-sea biology. It's a proxy for astrobiology. It provides a blueprint for how life might begin and sustain itself in some of the most common environments in the universe. What other forms of energy, what other strange chemistries might life have harnessed in places we haven't even dreamed of looking? Section 10. Xenophyophores. The single-celled architects in the soft, silty plains of the trench floor, you might see strange, delicate, lattice-like structures. They look a bit like flattened sponges or discarded nests. You might assume they are some kind of deep-sea plant or coral. You would be wrong. These are xenophyophores, and they are one of the strangest life forms imaginable. Each one of these complex structures, which can be up to 20 centimeters across, is a single cell. They are protozoans, a type of amoeba, and are the largest known single-celled organisms on Earth. To build their intricate homes, called tests, they collect minerals and particles from the surrounding sediment and glue them together with a natural cement. Why do they do this? Their massive branching structures dramatically increase their surface area, which is likely a strategy for absorbing the very dilute nutrients from the water and sediment. But they are more than just passive absorbers. They are ecosystem engineers. Their complex three-dimensional tests provide crucial habitats, tiny apartments and hiding places, for other, smaller organisms like isopods, worms, and crustaceans. In the otherwise flat, featureless expanse of the Hadal floor, a patch of xenophyophores is a thriving metropolis, a biodiversity hotspot. They create structure where there is none, What is truly mind-boggling is their resistance to their environment. They thrive in the cold and darkness, of course, but they also show an incredible resistance to heavy metals. The sediment they use for construction contains high levels of lead, mercury, and uranium. Yet the xenophyophores seem to sequester these toxic materials in their tests without being harmed. They are living archives of the local geochemistry. How does a single cell without specialized organs or tissues manage such complex feats of construction, chemical engineering, and ecosystem management? The xenophyophore challenges our very notion of what it means to be a simple organism. It reminds us that in the deep sea, the solutions to life's problems can be both elegantly simple and profoundly alien. Section 11. The language of the dark. Bioluminescence. As we journeyed down, we left the sun behind. But that does not mean the Hadal Zone is a world devoid of all light. It is a world filled with a different kind of light. A cold, living light produced by the creatures themselves. Bioluminescence. This is not a rare trick in the deep sea. It is the dominant language of communication. It is produced by a chemical reaction, usually involving a compound called luciferin and an enzyme called luciferase, and it serves a dazzling array of purposes. For predators, it can be a lure. The classic example is the anglerfish, dangling a glowing esca in front of its tooth-filled mouth to attract curious prey in the absolute darkness. For prey, it can be a defense. The vampire squid, when threatened, can eject a cloud of sticky bioluminescent mucus, a confusing cloud of light that allows it to escape. Some shrimp can spew a bright fluid from their mouths, a flash of light that temporarily blinds an attacker. Another defensive strategy is the burglar alarm. A small creature, when caught by a predator, will flash brightly. This doesn't help the creature itself, but it can attract an even larger predator, which might then attack the first one, allowing the smaller prey to escape in the ensuing chaos. Bioluminescence is also used for communication. Flashing patterns can be used to identify members of the same species, to attract a mate, or to signal territory in the vast, featureless dark. And for some, it is simply a form of camouflage. In the twilight zone, many creatures use photophores on their undersides to match the faint light filtering down from above, a strategy called counter-illumination that makes them invisible to predators looking up from below. While less common in the complete darkness of the trench floor, the principle remains. Life has invented its own light. It has created its own stars in the eternal night of the abyss. What does it say about the drive for life that in a world defined by a profound absence, it chooses to create its own suns, however small and fleeting? Section 12. A world of sound. We perceive the deep ocean as a silent world. It's a powerful image. The crushing pressure, the eternal darkness, the profound and heavy silence. But it isn't true. The Mariana Trench has a soundscape, and it is a bizarre and fascinating one. Sound travels much more efficiently in water than in air, about 4.5 times faster, and over much longer distances. The deep ocean is, in many ways, a world defined by acoustics. When scientists first lowered hydrophones, underwater microphones, into the Challenger Deep, they weren't sure what they would hear. What they recorded was a strange mix of the familiar and the alien. They heard the sounds of earthquakes, the low, powerful rumbles of the tectonic plates grinding against each other, the sound of the trench itself being born. They heard the distant moan of baleen whales, their calls traveling hundreds, even thousands of miles through the deep sound channels of the ocean. They heard the sounds of ship propellers from the surface, a reminder of the human world so far above. But they also heard sounds they couldn't explain. In 2016, researchers recorded a bizarre 5-part call that swept through frequencies from deep moans to a high-pitched metallic finale. They nicknamed it the Western Pacific Biotwang. After analysis, they believe it is a new, never-before-recorded type of baleen whale call. But its purpose and exact source remain a mystery. What about the creatures of the trench itself? Do they make noise? We don't fully know. The clicks and whistles of dolphins and toothed whales certainly penetrate the deep. The snapping of shrimp claws, the crunch of a crustacean's shell in a predator's jaws. These are all part of the acoustic environment. But for creatures without air-filled sacs to vibrate, making noise is difficult. Perhaps communication is not sonic but chemical or visual through bioluminescence. Or perhaps there are sounds we simply haven't learned how to listen for yet. Imagine you are a creature living there. Your world is not primarily one of sight, but of pressure waves, of vibrations, of chemistry, and of sound. How would you perceive reality? What would a thought feel like in a mind evolved for that kind of sensory input? Section 13, the Challenger Deep, a specific place. We often talk about the Mariana Trench and the Challenger Deep interchangeably, but it's important to remember that the Challenger Deep is a specific place. It is a relatively small, slot-shaped basin at the southern end of the much larger trench. It is the Mount Everest of the abyss, but unlike a mountain summit, it is not a single point. It's a basin about 11 kilometers long and 1.6 kilometers wide, comprised of 3 deeper pools that have been mapped by high-resolution sonar. The deepest of these, the Eastern Pool, is where the record-setting depths of nearly 11,000 meters have been recorded. What does it actually look like down there? The images and videos brought back by James Cameron and robotic landers paint a stark picture. The ground is not rock, but a fine, pale, almost white sediment. It's the accumulated remains of billions upon billions of tiny marine organisms, plankton with shells made of silica or calcium carbonate that have rained down as marine snow over millennia. This fine silt is easily disturbed, and any landing craft kicks up a cloud that can take hours to settle in the still water. It is a soft, undulating, almost desert-like landscape. There are no grand canyons or dramatic rock formations here at the very deepest point. It is a landscape of subtlety, but it is not lifeless. The sediment is covered in the tracks and burrows of unseen organisms. The ghostly snailfish drift by. Swarms of amphipods are attracted to the promise of food from a visiting lander. Strange, stalk-like foraminifera stand like tiny trees in the silt. It is a place of profound stillness. There are no currents to speak of. The water temperature is a stable, frigid 1 to 4 degrees Celsius. It is a world of constants. Think about the time scale of this place. The sediment you see is a history book, with each layer representing an era in the ocean above. Place this stable, this isolated, could it be a time capsule? Could the unique microbes living in that sediment hold clues to the ancient history of Earth's oceans, or even the origins of life itself? Section 14, the human signature. Our trash at the bottom, for all its remoteness, for all its alien nature, the Mariana Trench is not untouched by humanity. This is perhaps the most sobering and important discovery of the modern era of Hadal exploration. In 2019, explorer Victor Vescovo, piloting his submersible The Limiting Factor, was exploring the floor of the Challenger Deep when his camera settled on a stark, angular shape half buried in the pale silt. It was a plastic bag. Alongside it, candy wrappers. On other dives, they have found other debris. It's a gut-wrenching realization. We have managed to pollute the most inaccessible location on our planet. And it's not just macroscopic trash. The problem is far more insidious. Scientists have taken samples of the giant amphipods, the Alacella, from the trench. When they analyzed their fatty tissues, they found them to be riddled with human-made industrial chemicals, PCBs, polychlorinated biphenyls, which were banned decades ago, were found at concentrations 50 times higher than in crabs from one of China's most polluted rivers. How is this possible? These chemicals do not break down. They enter the food web at the surface, are absorbed by plankton, which are then eaten by small fish and so on. As they move up the food chain, they become more and more concentrated. When these animals die, their contaminated bodies sink, becoming part of the marine snow that feeds the deep. The trench, as the ultimate sink for the ocean, becomes a final, concentrated repository for our most persistent poisons. This discovery forces us to ask some difficult questions. We think of pollution as a surface problem, oil spills, the Great Pacific Garbage Patch. But this proves there is no away. Every single thing we produce, every chemical we release becomes part of a global system. The ocean connects everything. The fact that a plastic bag can reach the bottom of the trench before a human does is a powerful and shameful symbol. What does it mean for us to be explorers of a new frontier when we find our own garbage already waiting for us? Does this change our responsibility, not just to protect these environments, but to fundamentally rethink our relationship with the planet as a single, interconnected system. Section 15. The search for the origin of life. The Mariana Trench is not just a place of bizarre creatures. It's a laboratory for understanding one of the most fundamental questions in all of science. How did life begin? One of the leading theories for the origin of life on Earth is that it didn't start in a warm little pond, as Darwin speculated, but in the dark, energy-rich environment of deep-sea hydrothermal vents. These are fissures in the seafloor where seawater seeps down, is heated by magma, and then erupts back out, laden with dissolved minerals and chemicals. In the Mariana Trench, we find a specific and very special type of vent system. They are not the scorching black smokers. Instead, they are a result of that process we've mentioned before, serpentinization. As seawater reacts with mantle rock exposed by the subduction process, it produces hydrogen, methane, and simple organic molecules like hydrocarbons. The water that emerges is not superheated, but warm and highly alkaline. These alkaline hydrothermal vents create a unique chemical environment. They produce mineral-rich chimneys with tiny pores, creating a gradient between the alkaline vent fluid and the slightly acidic seawater. Why is this so exciting? Because those conditions are a near-perfect match for what many scientists believe is required to kickstart life. The mineral pores act as tiny concentrated test tubes. The chemical gradient between the vent fluid and the ocean is a form of energy that could power the first metabolic reactions. The hydrogen and methane are the raw building blocks for more complex organic molecules. It's possible that on the early Earth, vents just like these were the crucibles where inanimate chemistry first crossed the threshold into biology. By studying these vents in the trench today, we are essentially looking at a working model of a potential origin of life engine. Could it be that the answers to where we all came from are hidden not in the stars, but in the deepest, darkest place on our own planet? Section 16. The future of Hadal exploration. What does the future hold for our exploration of the trench? The era of the heroic one-off human dive may be giving way to a new paradigm. Persistent presence. The next great leap will not be about going deeper, but about staying longer and seeing more. This is the realm of autonomous underwater vehicles, AUVs, and advanced robotics. Imagine a fleet of small, intelligent, pressure-resistant drones released into the trench. They wouldn't need a multi-million-dollar support ship on the surface. They could be programmed to work together, mapping the seafloor in unprecedented detail, taking coordinated samples, and tracking organisms for days or weeks at a time. These future explorers will be equipped with advanced sensors, not just cameras, but DNA sequencers that can analyze microbial life on the spot. They'll have chemical sensors that can taste the water for signs of life or geological activity. They'll use AI and machine learning to make decisions on their own, to recognize something interesting, and investigate it without waiting for a command from a human pilot 11,000 meters above. This will allow us to move from taking snapshots of the trench to creating a long-term, high-definition movie. We could finally answer questions about the life cycles of Hadal creatures. How do they reproduce? How fast do they grow? How do they interact? But with this new capability comes new ethical questions. As we gain greater access, our potential for disruption also increases. How do we explore without contaminating? How do we ensure that the trench doesn't become a target for deep-sea mining, with companies looking to exploit the rare earth minerals that may be concentrated in the trench's unique geology? The future of Hadal science is a race between our curiosity and our wisdom. How will we choose to use this newfound ability to access the planet's last greatest secret? Section 17. Lessons in adaptation. The life forms of the Mariana Trench are more than just biological curiosities. They are living masterclasses in adaptation. They offer profound insights into the fundamental limits of life and the incredible flexibility of evolution. Consider the snailfish and its use of TMAO. This is a lesson in molecular engineering. By understanding how TMAO stabilizes proteins under pressure, biochemists could potentially design new methods for preserving sensitive biological materials, like organs for transplant or delicate pharmaceuticals. Without refrigeration, the pressure itself could become a tool for food preservation, or for driving chemical reactions that are difficult to achieve at the surface. Think about the chemosynthetic microbes. They are masters of energy efficiency, converting chemical energy into life with incredible skill. Studying their metabolic pathways could unlock new forms of biotechnology. We could harness these microbes or their enzymes to clean up pollution, to produce biofuels from waste products, or to develop new industrial processes that don't rely on harsh chemicals or high temperatures. They provide a blueprint for a different kind of industry, one based on the elegant solutions of deep-sea life. Even the giant amphipods have lessons to teach us about surviving in a low-nutrient environment. Their slow metabolism and efficient energy storage are a marvel. In an age where we are exploring long-duration space travel, understanding how to put life into a low-power, sustainable state is of critical importance. The solutions found in the most extreme environment on Earth could hold the keys to helping us survive in the most extreme environment off of it. When we look at the trench, what should we see? A barren wasteland? Or a treasure trove of biological genius, with 3.8 billion years of research and development just waiting to be understood? Section 18, the psychological oceanlet, is step back from the science for a moment and consider the trench as a metaphor. It exists within us as much as it does in the Pacific Ocean. Carl Jung spoke of the shadow self, the deep, unconscious parts of our own minds that are hidden from the light of our daily awareness. The ocean, and particularly the deep sea, has always been a powerful symbol for this inner world. The journey to the bottom of the trench is a journey into the unknown, a confrontation with the alien, the dark, and the primordial. Why are we both terrified and fascinated by it? The fear is easy to understand. It is the fear of being crushed, of the dark, of drowning, of the unseen monster. It represents a total loss of control, a submission to a force of nature so much greater than ourselves. But the fascination is just as powerful. It's the allure of the mystery, the promise of discovery, the hope of finding something new, not just about the world, but about the nature of life itself. The explorers who descend, whether in person or by robotic proxy, are not just collecting data. They are on a quest. They are modern-day Argonauts, journeying to a golden fleece of knowledge hidden in the underworld. When you think about the vast, dark, pressurized space of the trench, what comes to your mind? Do you feel a sense of claustrophobia, of being trapped? Or do you feel a sense of wonder, of infinite possibility in that darkness? Our reaction to the deep sea says a lot about our own psychology, our own relationship with the unknown. It is the ultimate Rorschach test, a dark, watery ink blot on the surface of the planet, onto which we project our greatest fears and our most profound curiosities. What do you see in the abyss? Section 19, the interconnected planet. If there is one single, overarching lesson from our journey to the Mariana Trench, it is this: there is no part of our planet that is truly separate. We used to think of the deep ocean as a world apart, isolated and insulated from the surface. We now know that is fundamentally untrue. The food web of the trench is powered by the death of plankton in the sunlit waters thousands of meters above. The chemical composition of its creatures is dictated by the pollutants we release into our rivers and atmosphere. The soundscape of the abyss is punctuated by the passage of our ships and the calls of whales that migrate across the entire globe. The trench is connected to the surface by gravity, by chemistry, and by the global circulatory system of the ocean currents. In turn, the deep ocean has a profound impact on the world we live in. The immense volume of the deep sea acts as a massive heat and carbon sink. It absorbs a huge portion of the excess heat and carbon dioxide we are pumping into the atmosphere, buffering us from the full effects of climate change. The processes happening in the deep, including the subduction in the trench, are part of the long-term carbon cycle that regulates the planet's climate over geological timescales. We are part of a single, complex and deeply interconnected system. An action on a farm in the American Midwest can, through a long and complex chain of events, eventually have a measurable impact on the chemistry of the sediment at the bottom of the Challenger Deep. This is a staggering thought. It imparts a sense of immense responsibility, but also a sense of profound wonder. We live on a planet that is not a collection of separate environments, but a single, living, breathing whole. Doesn't this knowledge change how we view our place in the world? We aren't just living on the Earth. We are living in it, a part of its global metabolism. Section 20. The return to the surface, our journey is now coming to an end. We begin our ascent, rising up from the Hadal zone, out of the eternal night of the abyss. We pass back through the midnight zone, where the only stars are the living lights of the creatures who call it home. We rise further into the twilight zone, and for the first time in our journey, we see it, a faint, impossibly distant blue glow. It's the sun. As we rise, that glow brightens from a dim ghost to a shimmering ceiling. The pressure lessens its grip. The temperature begins to rise. We breach the 200-meter mark and burst back into the epipelagic zone, the world of light, warmth, and color. We break the surface, returning to the world of air, of weather, of sky. But we are not the same as when we left. We have been to the most extreme location on our planet. We have witnessed how life, in its relentless drive to exist, has conquered the challenges of absolute darkness and unimaginable pressure. We have seen that the deepest, most remote place on Earth is not a dead zone, but a dynamic living system, filled with strange and beautiful forms of life. We have learned that it is not separate from us, but is intimately connected to everything we do. The true purpose of exploration is not just to see what is there, but to return with a new perspective. The Mariana Trench holds up a mirror to us. It reflects our ingenuity in the vessels we build to visit it. It reflects our carelessness in the pollution we have sent there ahead of us. And it reflects our own biology back at us, showing us the fundamental chemical and physical rules that life must obey, and the infinite creative ways it has found to bend them. The next time you look out at the ocean, don't just see a flat blue surface. See the depth, see the layers of darkness, the pressure, the cold, and see the life that persists there against all odds. And ask yourself, what other impossibilities have we yet to discover, both in the world around us and within ourselves? We speak of the Trieste in 1960 as the dawn of Hadal exploration, the moment humanity first touched the bottom. But the human desire to peer into the deep, dark ocean did not begin with a vessel capable of reaching its ultimate floor. The psychological barrier, the transition from dredging the depths with nets to actually sending a human consciousness into the crushing dark, was broken decades earlier. To truly appreciate the technological marvel of visiting the Challenger Deep, we must first journey back to the 1930s and join two pioneers, William Beebe and Otis Barton, as they prepared to descend in a machine that was, by modern standards, terrifyingly primitive, the bathysphere. Imagine a hollow steel ball, just under 5 feet in diameter, with walls 1.5 inches thick. It had 2 tiny fused quartz windows, each 3 inches thick, the strongest transparent material known at the time. This sphere had no engine, no propellers, no way to steer. It was, in essence, a cannonball with windows, designed to be dangled at the end of a colossal steel cable from a ship on the surface. Inside, Beebe, a naturalist, and Barton, the engineer who designed it, would be sealed into the cramped, cold space. Their only connection to the world of air and light was a single electrical cable for a searchlight and a telephone line. If that cable snapped, the bathysphere would become their tomb, plummeting to the seafloor with no hope of recovery. Think about the sheer audacity and the raw courage of that endeavor. Their goal was to descend a half-mile down, about 800 meters, into the twilight zone. The pressure there would be immense, though a fraction of what exists in the Mariana Trench. Every descent was a gamble against catastrophic failure. The sphere was sealed with a massive hatch held on by 10 heavy bolts. Once sealed, they were inside for hours. Oxygen was supplied from a tank, and trays of soda lime were used to absorb their exhaled carbon dioxide. Another tray of calcium chloride was there to absorb moisture, as the cold metal walls would cause their breath to condense and literally rain down on them. On their record-breaking dive in 1934, Beebe became the first human biologist to witness the deep-sea ecosystem firsthand. His descriptions, relayed up the crackling telephone line, are pure poetry. He spoke of constellations of unknown bioluminescent creatures drifting past his tiny window. He described fish with glowing lures, shrimp that shot out clouds of luminous fluid, and terrifying fang-toothed predators emerging from the blackness into the beam of his searchlight. He was a Columbus of the vertical frontier. Before him, deep-sea life was a collection of dead, colorless specimens hauled up in nets, their bodies often mangled by the pressure change. Bibi saw them alive, in their own world, a world of living light. Why is this story so important to our journey to the Mariana Trench? Because Bibi and Barton proved that the deep was not a lifeless, barren void. They showed it was a vibrant, active, and fantastically strange world. They ignited the public imagination and inspired the next generation of engineers and explorers. Their work highlighted the fundamental limitation of their approach, the tether. Being tied to the surface meant they were at the mercy of the ship's movement. A rolling wave could send them jerking up and down on the cable. It was clear that to go deeper, to reach the true Hadal depths, humanity would have to cut the cord. This is what led to the bathyscaphe, like the Trieste, a free-swimming vessel, an underwater airship. The bathysphere was a chained observer. The bathyscaphe was a true explorer. Does this progression from a tethered ball to a free vehicle mirror our own development? From being tied to our immediate surroundings to venturing out into the unknown, first with tethers, and then finally setting ourselves free. We have spoken of the microbial kingdom that reigns in the Hadal mud, the bacteria, and archaea that form the foundation of this unique ecosystem. We see them as the bottom of the food chain, the ultimate recyclers and chemosynthesizers. But this view is incomplete. For wherever there is a thriving population of bacteria, there is another, even more numerous and mysterious entity that preys upon them: viruses. Specifically bacteriophages or phages, viruses that exclusively infect bacteria, and the deep sea, including the Mariana Trench, is teeming with them. If you were to take a single milliliter of Hadal sediment, you would find up to 10 billion viral particles. They outnumber microbes by at least 10 to 1. This is the most abundant biological entity in the trench, and yet, we are only just beginning to understand its role. What are these deep-sea viruses doing? They are not passive players. They are the primary predators and puppet masters of the microbial world. Their entire existence is a cycle of infection and replication. A phage will attach to a bacterium, inject its genetic material, and hijack the cell's machinery, forcing it to produce hundreds or thousands of new viruses. Eventually, the bacterium bursts open, a process called lysis, releasing the new viral swarm to find new hosts. This is a constant microscopic cull. Every single second in the trench, it is estimated that viral infections are destroying up to 20% of the entire microbial population. This process is known as viral lysis, and it has enormous consequences. When a bacterium is lysed, all of its internal, nutrient-rich contents, proteins, lipids, carbohydrates, are released into the surrounding environment. This creates a dissolved soup of organic matter that can then be absorbed by other non-infected microbes. This is called the viral shunt. It prevents nutrients from being locked away in microbial bodies and passed up the food chain to larger organisms. Instead, it keeps those vital nutrients recycling at the very lowest level of the ecosystem. So are these viruses a destructive force, or are they the engines of nutrient cycling, ensuring the entire microbial community remains dynamic and well-fed in a world of scarcity? But their influence is even more profound. Viruses are major drivers of evolution. By constantly preying on the most successful and abundant bacterial strains, a concept known as the kill the winner hypothesis, they prevent any single species from dominating the ecosystem. This opens up ecological niches for rarer microbes to thrive, thereby promoting and maintaining biodiversity. Furthermore, viruses can sometimes carry genes from one host to another in a process called horizontal gene transfer. A virus might accidentally package a piece of its host's DNA along with its own. When it infects a new bacterium, it can transfer that gene. This is a powerful evolutionary shortcut. A bacterium could, in an instant, acquire a gene for resistance to a heavy metal, or the ability to metabolize a new chemical, without having to evolve it over thousands of generations. The viral population of the trench could be a massive shared genetic library for the entire microbial community. They are not just predators. They are genetic couriers, accelerating the pace of adaptation in this extreme environment. What does this reveal about evolution? We think of it as a slow, gradual process. But here, in the dark, could it be a world of sudden leaps forward, all brokered by these invisible viral architects? We've seen the incredible physical and biochemical adaptations of Hadal life, the cartilaginous skeletons, the piezolyte molecules like TMAO. But these are just the outward expressions of something much deeper, an entirely different genetic blueprint. To truly understand how to build a creature that can thrive at 100 atmospheres, we have to look at its DNA. The instruction manual for life in the trench has been edited, revised, and annotated by the relentless pressure over millions of years. And what it reveals is fascinating. When scientists sequence the genomes of Hadal creatures, like the Mariana snailfish or deep-sea amphipods, they find unique signatures of adaptation. One of the most common is gene duplication. Imagine a critical gene that produces an essential protein. In a surface animal, there might be just one copy of this gene. But in a deep-sea animal, scientists often find multiple copies. Why would this be useful? Think of it as having backups of a critical file. The immense pressure can physically damage DNA molecules and disrupt the process of reading the genetic code. Having multiple copies of a key gene ensures that even if one copy is damaged or misread, the cell can still produce the protein it needs to function. But it's more clever than just having spares. This duplication also allows for evolutionary innovation. With multiple copies, one copy can continue its essential day-to-day job, while the other copies are free to mutate and experiment. One of these mutated copies might produce a slightly altered protein that works even better under high pressure. Over generations, this new optimized gene is selected for, and the organism becomes even better adapted. We see this in the genes responsible for building the very proteins that TMAO protects. The snailfish doesn't just have more TMAO, it has evolved proteins that are inherently more stable and compact, making them less susceptible to being crushed in the first place. Another incredible genetic story is the loss of certain functions. Genes are costly to maintain and replicate. If a biological system is no longer needed, evolution is ruthless about discarding it. For instance, many deep-sea creatures have lost the genes associated with vision and circadian rhythms. In a world of eternal, absolute darkness, the complex machinery for seeing color or for responding to a 24-hour day-night cycle is useless baggage. By deleting these now obsolete genes, the organism saves precious energy. But what have they evolved in their place? We see an expansion of genes related to chemosensation, the ability to smell or taste the water, and mechanoreception, the ability to sense tiny pressure changes and vibrations. They have genetically rewired themselves, trading a world of sight for a world of scent and touch. What does this tell us about our own genome? How much of our own DNA is a relic of a past we no longer inhabit? And what new genetic tools are we, as a species, slowly evolving to meet the pressures of our modern world? Close your eyes again. This time, try to imagine navigating your world not just for a moment, but forever. Imagine that your primary sense of sight is simply gone, a forgotten relic. How would you perceive reality? For the creatures of the Mariana Trench, this is not a thought experiment. It is their existence. They live in a sensory landscape that is almost incomprehensibly different from our own. A world built not of images and light, but of pressure waves, chemical trails, and perhaps even electrical fields. Let's start with touch, or more accurately, mechanoreception. Many deep-sea fish, including relatives of the snailfish, have a highly developed lateral line system. This is a series of pores and canals running along the side of the fish's body, containing tiny hair-like cells that can detect minute movements and vibrations in the water. In the utter stillness of the trench, this system becomes a form of long-distance sight. The fish can feel the pressure wave of a struggling shrimp many meters away. It can sense the subtle displacement of water caused by another predator approaching from behind. It builds a 3D image of its surroundings made entirely of water dynamics. It can literally feel the shape of the dark. Then there is the world of chemistry. The water of the trench is not a uniform fluid. It is a tapestry of chemical information. Scavengers like the giant amphipods are, in essence, mobile tongues. Their antennae are covered in exquisitely sensitive chemoreceptors, They are tuned to detect the specific molecules released by decaying flesh, so-called necromones. A single molecule of a decomposing carcass can trigger a response, allowing them to home in on a food fall from kilometers away, navigating by following a concentration gradient that is imperceptible to us. For them, the world is a landscape of smells. A whale fall isn't an event. It's a sudden fragrant bloom in the silent dark, a beacon of scent that screams life. And what about senses we barely possess at all? Some deep-sea creatures, particularly sharks and their relatives that live in the abyss, are masters of electroreception. They have specialized organs called the ampullae of Lorenzini that can detect the tiny bioelectric fields generated by the muscle contractions of other living animals. Even a creature buried in the mud is not hidden from them. Its own heartbeat and respiration create a faint electrical aura that the shark can see as clearly as we see a light bulb. Could this sense exist in the trench's invertebrates? We don't yet know, but it forces us to reconsider what it means to be hidden. In a world without light, life has evolved other ways to make itself known. Which of our own senses, if amplified 1,000-fold, would completely change our perception of reality? What if we could feel the subtle vibrations of the air, or smell the emotional state of another person? The creatures of the trench live in such a world, a testament to the fact that our human sensorium is just one of many possible ways to experience the universe. We have touched upon the idea of the interconnected planet, how the surface feeds the deep. But this connection is not a one-way street. The Mariana Trench is not just a passive recipient of the world's detritus. It is an active and profoundly important player in the long-term health of our planet's climate. It functions as the ultimate carbon coffin. To understand this, we need to think on a geological timescale, a scale of millions of years. The carbon that makes up all life, from plankton to blue whales to us, is part of a global cycle. It moves between the atmosphere, the oceans, and the land. One of the biggest questions in climate science is, where does carbon go to be removed from the system for a very, very long time? The trench is one of the answers. We've talked about marine snow, the slow rain of organic matter. This material is rich in carbon, pulled from the atmosphere by photosynthesis at the surface. When it reaches the Hadal floor, some of it is consumed and recycled by the ecosystem, but a significant portion gets buried in the sediment. Layer upon layer, year after year, this carbon-rich mud accumulates. It is a slow, patient process of sequestration. But what makes the Mariana Trench unique is the next step: subduction. That sediment, full of trapped organic carbon, is on the Pacific Plate, that geological conveyor belt that is plunging down into the Earth's mantle. The trench is literally swallowing the seafloor. And as it does, it takes all that accumulated sediment and all that trapped carbon with it. It is pulling carbon out of the biosphere and injecting it deep into the planet's interior, where it will be locked away for geological epochs. It is one of the planet's most effective and permanent carbon sequestration mechanisms. The biological pump, driven by life at the surface, sends carbon down. The geological pump, driven by plate tectonics, buries it forever. This process has been happening for millions of years, acting as a long-term thermostat for the Earth, helping to regulate the amount of carbon dioxide in the atmosphere. So, when we look at the trench, we should not just see a biological wonder, we should see a critical gear in the planetary machine. However, this system is now under threat. A warmer, more acidic ocean, a result of our own atmospheric carbon emissions, can disrupt the plankton at the surface that form the base of this entire process. We are altering the very first step of a system that is, ironically, helping to protect us from our own excesses. By overwhelming the ocean's capacity to absorb carbon at the surface, are we effectively clogging the drain at the bottom of the world? Understanding the Hadal zone is therefore not just about satisfying curiosity. It is a crucial part of understanding the Earth's climate system as a whole. It reminds us that the health of the very surface we live on is inextricably linked to the slow, powerful, and utterly invisible processes happening in the deepest parts of our world. We send our robotic landers and our human-occupied subs down into the crushing dark, and they return with breathtaking images and data. But the ultimate goal of biological exploration is not just to see, but to touch, to collect, to bring back a piece of that alien world for study in our labs. And this is where one of the greatest technical challenges lies: the agony of the ascent. For the creatures of the trench, the crushing pressure isn't a hostile force. It's a life-sustaining embrace. Removing them from it is as violent and destructive as throwing one of us into the vacuum of space. This is the paradox of Hadal sampling. How do you study a creature when the very act of retrieving it destroys it? Imagine a Mariana snailfish. Its cells are filled with piezolites like TMAO, molecules that are perfectly balanced to counteract the external pressure. As a submersible brings that fish up, the external pressure drops. For every 10 meters it rises, 1 atmosphere of pressure is removed. Inside the fish's cells, the internal pressure now catastrophically outweighs the external pressure. Cell membranes, no longer compressed into stability, begin to stretch and tear. Proteins that were held together by the pressure begin to unravel. Dissolved gases in its tissues expand, forming bubbles. By the time it reaches the surface, the animal is not just dead. Its tissues have been ripped apart on a molecular level. It arrives as a gelatinous, shapeless wreck. To overcome this, scientists and engineers have developed incredible tools called pressure-retaining samplers. Think of them as miniature, robotic versions of the Trieste's cryosphere. A water sampler might be a strong titanium cylinder with a valve that closes at the bottom, trapping a sample of water, and with it, the microbes living there. It is then hauled to the surface, maintaining its internal pressure of 1,100 atmospheres the entire way. In the lab, this cylinder can be attached to other high-pressure chambers, allowing scientists to study the microbes under their native conditions. Similar devices have been designed to trap larger creatures like amphipods, giving us our first look at how these animals behave under pressure in a controlled setting. But this technology is incredibly difficult, expensive, and limited to small organisms. We still cannot bring a snailfish back alive, and even for microbes there is the issue of temperature. As the sampler is hauled up through thousands of meters of warmer water, the sample heats up, potentially killing the cold-loving piezophiles inside. Samplers must therefore be perfectly insulated. This highlights a crucial point. The trench is a multivariable environment. It's not just high pressure. It's high pressure, low temperature, specific chemistry, and total darkness. To truly understand its inhabitants, we must recreate their entire world in a box. So the next time you see a photo of a strange, pale creature labeled from the Mariana Trench, remember the extraordinary journey it has taken, and the immense technical ingenuity required to bring even a ghost of its true self back to our world. It forces us to ask, will we ever be able to truly understand an environment that is fundamentally lethal to us, and to which we are just as lethal?